Hi everybody. I am out here in nature today at, where are we? Petal River Park. And basically I just wanted to give you a visual guide as to how you're going to go around and make your iNat observations for part 1 of our semester-long project. So what I'm going to do is break it down into how you're going to make observations of fungi, plants, and different kinds of animals today. Obviously, we don't have time, and I'm not going to make a video that covers every single type of everything you might encounter, because I don't know where you all are. You might be in Mississippi, you could be in Europe for all I know. But hopefully this will give you an idea. If you have any questions later, of course you can feel free to email me or visit me during my virtual office hours. Yeah, iNaturalist is it's an app. Okay, generally it's mostly used on your phone. You can download it, it's super easy to use. But iNaturalist is a service that has almost 300 million observations of different organisms uploaded to it across the world. So what iNaturalist does is when you make an observation, you take a few pictures, make sure they're relevant structures and whatnot. It uses all of the photos that have been uploaded across the world to help you identify that thing. Again, not always correct, but generally pretty helpful. And this is actually a service that's very pro-scientist, and it's very citizen science driven. And scientists like myself have actually used it in research. So whenever you're making these observations of whatever tree you're looking at, whether like this oak here or the cross vine that's growing up this oak, you want to make sure that they're good observations so that you wouldn't feel bad if someone wanted to use them in their research. Okay? So yeah, iNaturalist is just a great resource. It's a great resource to look up ranges of organisms that have been observed across the world. It's a great resource to look up, try not to trip on roots here, to look up all the different variation that can occur across the range of these organisms. So make sure that your observations are really good, okay? Okay, so some ground rules before we get too far into this video. Number one, if I say something, I don't have a script right now, so if I say something that is incongruous with what is written in the guidelines that I posted on Canvas for you, go with what's written, not with what I say. I could misspeak and not even catch it right now. So just a warning there. Secondly, I want to make sure that you know that you are not allowed to go on private property to do this project unless you have permission, written permission from the owners of that property. And if you're unsure, don't do it. Stick to public areas and public parks. That's a great guideline. Let's see, what else am I doing here? Make sure you're aware of your surroundings when you're out here in nature. Obviously, we're in January right now, so there's not really much going on. We're going to look around for what we can find, but if you're doing this later in the semester, be aware of general things like poison ivy. It's not going to hurt you, but you know, some people can have a stronger reaction than others, so just be aware of that possibility. Venomous snakes really probably aren't going to be an issue in this first part of the semester. However, if you're in a warmer, more southerly area of the United States or elsewhere, be aware of what venomous snakes might be in your area. And be able to identify those. And we'll talk more about that when we get to how to identify a vertebrate later. And I think that'll do for now, as far as overall safety and guidelines. One other thing is just making sure that you're aware of the scope of this project. It's nothing too difficult. You have until, I believe, March 27th, Friday, March 27th, to log and complete 25 observations on iNaturalist. So in those observations, you need to make sure that they're as identified as they possibly can be using the iNat AI, I suppose. If you know what they are, for sure, you can identify them to species yourself. If you can, I encourage you to do that. If not, don't make up something, and don't trust that iNaturalist will get it right. They don't have to be necessarily correctly identified to count for this project. This is more just to get you aware of iNaturalist, but I will say that everything you observe for this first part of the project is going to dictate what you can do the second part of the project on. We'll talk more about those details about the second part of the project a little later in the semester. So you have to do 25 of these projects. They have to have multiple photos. You need notes on anything that an observer or that another user on iNaturalist might not be able to see or be aware of based on the photos that you took. So you need to make sure that you have detailed photos, that they're not blurry. Notes can be notes about the population that's around it. Is it by itself? How is it growing? Is it in a large population? Things like that. Yeah, and I think that's it. So the next thing we're going to do, is go around and give you some examples of types of pictures that you can make, and then later I'll go to my desktop and show you how we can annotate those with those notes and phenology observations. All right, so for this project, you can take pictures of whatever animals, plants, fungi, or lichens you want. There are just a few rules about what is and is not allowed. What's not allowed are things that are cultivated or horticultural, so that includes things like tomatoes, corn, crops in general, not going to cut it, and things like house plants or things that you plant in your yard like roses. Those are not native species, or those are horticultural variants that aren't real in nature. So those are things we have made. Another thing are domesticated animals. As much as I would love to see pictures of your dog, you can't upload those to iNat. They're domesticated, so no cows, chickens, goats. What are other things? Dogs, cats, parakeets, weasels, anything. If you own an animal, that's not going to cut it. No fish that you own either. They have to be in nature where you find them. And I think that does it. That'll do. All right, so this is a fungus. These are actually some basidiomycetes, if you've taken my botany class, or biology 2, which you probably have taken. These are a type of shelf fungus, so they just generally grow on dead trees, and they're very hard fungi. So that's what these are. I know what these are, which makes them a good example for me to show you here. So when we're making observations for iNaturalist, we want to make sure we have a few things. If I've said this already in another video, it doesn't matter. But we have to make sure that we take multiple photos. So we don't just want one photo of a log. The good news is about fungi is if you're seeing the fungi and it looks anything like this or like a mushroom, that is the reproductive structure. And usually that's the only thing showing on a fungus. So whenever I'm making observations of fungi, I need to be aware of a few things. One, a lot of fungi are poisonous. So don't pick or touch fungi and then lick your fingers. Bad idea. Another thing is that these are wild fungi. We want to make sure in our observations we're taking pictures of wild things. And whenever I'm taking pictures, I can just take them on the Notes app on my phone. Notes app? The camera app, that's what it's called. And so what I want to make sure I get whenever I'm taking pictures of fungi is a view of the top of the fungus. So close up in focus view. You can turn the live photo thing off. Don't need that. So a few photos of the top, not that I'll necessarily use all of those. And then fungi are safe to touch generally here in Mississippi. So I can pick one off and you see it stripped the bark off. You can see the inside, the actual body of the fungus all over that right there. So I have pictures of the top of it and it's kind of fuzzy on top. That might be a good thing to note in the notes under this observation. And then on the underside, I want to make sure that I get a good picture of the underside. So as close up and detailed as I can get from several different angles, because these are all characteristics that are useful to identify it to any more specific ranking. And then I'll just sit it back down right there, let it be happy. Yeah, and that's pretty much it for fungi. You just want to make sure you have pictures of the top of the cap and the bottom of the cap. And if it has a stipe or like a little stem that it stands on, you want to make sure you get good pictures of that as well. But again, we have a lot of poisonous fungi, especially the amanitas. So if you touch fungi, make sure you wash your hands and don't lick your fingers. Don't cook with hands that haven't been washed after touching fungi. Yeah, I think that'll do it. Okay, so we just found another shelf fungi and it's pretty cool because it's huge. This thing's more than a foot across, about 1.5 feet across. So just to show you again, one more example of how we observe fungi. We take a picture of the top, and then we want to make sure we get the gills underneath, which in this case are pores, because this is a polypore. So we'll just go under there. I'm going to click the side button to take that picture. And then I'm going to take a picture of the top with my hand present, so we have some sense of scale. There's actually another one right here. But yeah, it's just one more cool one. Okay, so I just flipped this log right here, and I got a really good salamander under there. So this is the first vertebrate we've run into. So I want to encourage you, or should I say discourage you, from handling vertebrates unless you have experience doing so, and any relevant certifications, because if you don't know what a vertebrate is, it could very well be poisonous, it could very well have teeth, it could very well be endangered for all you know. So I'm going to discourage you from handling vertebrates. This is a salamander. It's a type of amphibian. So how we want to take observations of a salamander is get my handy dandy camera out, and without disturbing it, get pictures of the top. And I generally like to keep taking pictures until it will let me get as close as I can, because I know what this is. This is a Mississippi slimy salamander. It's trying to hide. I want to get a lot of really good close-up pictures. These things are harmless unless you touch them. They release a compound on their skin that the reason they get their name slimy salamander is because when threatened, they'll release a compound on their skin that makes them very slimy. And whenever that compound touches air, it gets really sticky like glue. So they'll almost glue your hands together if you have them in your hand. So I'm not going to handle it. All right. So we are in the middle of winter here, so there's not just a whole lot of activity going on here when it comes to vertebrates. Other than salamanders, it's too early for snakes, it's probably too cold for turtles and whatnot. But one thing that is going to be out during the winter are warm-blooded things, so things like birds and mammals. Though mammals are really quick most of the time, you're probably not going to see many unless you do unless you go out of your way to observe them. You're probably not going to see a mammal. But what I do want to tell you is that a lot of the times you can find evidence of mammals, and that is reportable on iNaturalist. So if we look right here, we can see that there are some little poops that are here, and these aren't fresh, which is why I'm handling them, and I know generally what this is, so I'm not worried about this poop. Don't touch wet poop, because that's just gross. But these little poops right here are evidence of a mammal, and mammals often have very specific digestive tracts, which makes their poops pretty identifiable a lot of the time. It's a good clue. So if you do see poop, in the woods, not your own poop, you can take a picture of it and report it on iNaturalist and see if it can help you. So this is probably either a rabbit, it probably is a rabbit, but could be a deer as well, a small deer. So I can take pictures of that, put it on iNaturalist. Yeah. Another group of animals that you're probably going to you might be inclined to take pictures of and record on iNaturalist, and I would encourage you to if you can, is a notoriously difficult group to photograph, and those are birds. So I don't know if you can hear them around me right now, but there are some small chickadees going off around here. iNaturalist does let you record sound and upload sound, so that's very important, especially for bird ID. A lot of you probably know more about birds than I do, having taken avian with Dr. Phillips. But yeah, you can try to. You can even try to get photos of them. They're very difficult to take photos of if you don't have a very good camera, so do not try to upload blurry photos of birds, because that's not very useful generally. What you want to, or a good opportunity to get pictures of birds is on campus or in public parks, because they're generally more used to people than birds in the woods are. If you go down to ponds or lakes or rivers, a lot of times you can find waterfowl, like ducks and coots and whatnot. Yeah, am I missing anything? Oh, there's a woodpecker right there. So the group that I probably haven't talked much about so far are the non-avian reptiles, so things like alligators, turtles, lizards, and snakes. I've not talked about them much because that's probably going to be the least common thing you'll see in the early spring, because it's going to be too cold. You might see turtles, and if you see a turtle, that's fine. Turtles are generally harmless if you don't handle them. They're completely harmless if you don't handle them, actually. All of these things are. the thing I want to make you aware of are snakes, because even though we have about 50 to 60 species of snakes in Mississippi, 6 of them are venomous, and they will all, if you get bitten, get you put in the hospital. So no handling snakes. Again, no handling vertebrates, but especially no handling snakes. I don't know whether or not you've taken herpetology. That doesn't matter for the purposes of this class. You are not allowed to take pictures of you handling vertebrates. Yeah, so that'll do. Okay, it just occurred to me, because we're by the river, that the only group that I didn't talk about was fish. Fish are fine. You can take pictures of fish if you can put your camera or phone in water, I suppose. That's perfectly fine with me. Yeah, that's fine. Again, you're not allowed to handle vertebrates for this project, so no handling the fish. But if for whatever reason you can get identifiable pictures of a fish in the water, that's fine. That'll do. So this is an example of one that you don't want to handle. This is a true centipede. You can tell it's got only 1 pair of legs per body segment. And what you can't see in this video is that on the sides of its head, it has 2 venomous spurs, and there it goes. So you want to make sure you don't touch those, because they will bite you and it will hurt. But you can see all of this nice mycelia hiding under this log that's helping it rot. Good fungus log right here. So right here we've actually got a larvae of a type of insect. I think it's a beetle. Oh, and there's a centipede right there creeping around. Again, we want to make sure we don't touch, because I'm not trying to, get my finger stung right now. But these larvae are generally harmless. They can't do much. They also only have a few legs, so they're not a centipede or a millipede. So they're not harmful. You can try to observe that. iNaturalist sometimes is okay identifying larvae. Adults are better though. So actually under that same log as this slimy salamander over here, we found what's called a best bug, which are generally harmless. I don't really want to get bit by it, so I'm not going to do too much, but they are harmless little beetles. Again, she's kind of putting on a display right now. But if I'm taking pictures, I want to make sure I get the underside of it as well as the top side. Do not bite me. We'll flip it back over for that. Yeah. So, you can get good pictures of this, but especially beetles, because they're so diverse, iNaturalist might have a hard time identifying it. That one shouldn't be an issue because that's an extremely common species. Yeah. Cool log. So whenever we flip the log back over, we want to make sure that we're not crushing anything, so we're going to do it very carefully like I showed you earlier. And just like that. Boom. Done. Okay, so functionally when you're in nature and you're looking for plants to observe, there are a few characteristics that you want to make sure that you have. So one of those is reproductive structures. So especially if you've taken my botany class, you know that reproductive structures kind of are very important for determining what plants are what. So whether that's a flower, could possibly be flower buds, though that's not generally as useful, or fruits, or cones if you're looking at a gymnosperm, or sporophytes if you're looking at, small mosses or liverworts, or sori that might be on the underside of fern fronds. Whatever group of plants you're looking at, you want to make sure that, if at all possible, that you have reproductive structures. So this example I'm going to show you here is a terrible example of a plant that you should observe. So we can move in a little bit closer. This is, I'll give some of you guys that have taken some upper level plant classes a moment to guess. But this is Florida anise. So Florida anise is an early diverging eukaryotic plant. And as you can tell, they're really just sticks with leaves on them right now. There are no flowers to be found at all. So this is not the ideal specimen to observe, at least at this moment. It will flower this spring, and you'll be able to come out here and take pictures of these big, beautiful red flowers. What I'm showing you this for, though, is that there are other features of this plant that I'll experience here. You still won't be able to experience that in the video that, you know, if you were observing this plant, you would want to make sure that you point out in the notes section of your observation. So, you know, I might pick a leaf off of it. You want to use all your senses when you're trying to observe things. So this is a plant where you can actually crush up the leaf. It's got a very leathery kind of leaf. So I can tear the leaf and it smells so good. So it smells very licoricey. That's why it's star anise, just like, or it's related to star anise. It's Florida anise. So when you crush up the leaves, they release this chemical. Smells great to me. A lot of people hate it. But that chemical smell might be something that you want to describe in the notes section of this observation. So another thing you might want to observe is the population. So we have one here, and then we have another one here, and then we have some more right here, all over the place, even way down here, the tiny little seedlings. You can describe the population structure in the immediate area. So you can say occurs in a patch, or like you can estimate there might be 30 individuals here in this small area. That can be very helpful information for other people that look at your iNaturalist posts. All right, so like I just mentioned in that last video, we want to make sure that we get reproductive structures on plants. That was kind of a small shrub. When we get even smaller, like these tiny little mosses right here, we can go down here. Taking a picture of just a plain green moss generally isn't that helpful. What is helpful is if you find a moss that has these structures that are standing up out of it. If you've had Bio 2 or Botany Labs, you know that this structure is called the sporophyte of the moss, and all this green little feathery stuff at the bottom is the gametophyte, that is the haploid tissue. This is the reproductive structure that you really want to take a picture of, because these are often distinctive among species, so distinctive shapes. So if you do take a picture of a moss or a liverwort, you want to make sure that this is in those pictures. And again, you get as close as you can. You can often include like a dime or your fingernail in that for scale, so that people in the can tell how big these are in your photos. Okay, so you guys, really exciting moment for me this year. Best day of 2026 so far. We just found the first flowering wild violet of the year. So this is actually a great example because we haven't done a flowering plant when it's flowering so far. So to show you guys what kind of pictures you want to make sure you have when it comes to plants writ large, but also specifically flowering plants. So you can see we have the reproductive structures right here. I didn't ask you. So we've actually got a flower bud way down here. We can pull in on that. We got a flower bud right there, and then we've got two more mature flowers. This one's about to start wilting. This one's already wilted. So when we're taking pictures, we can go to our handy dandy photo or whatever camera app. I've already messed that up once. Oh well. Moving on, we're going to make sure that we get pictures of the top of the leaves. So make sure we get pictures of the petiole or the leaf stem right here. One other thing we want to make sure we get is the bottom of the leaf. Okay, so that deals with the sterile structures. Now what we want to make sure we get a picture of are the other parts of this flowering plant, I suppose, which are the reproductive structures. So since we have a flower bud right here, down here at the bottom, it hasn't quite gotten tall yet. I'm going to make sure I get a good side, close-up side profile picture of that. I'm going to make sure we get one of that. We're going to go get a picture of the front of this good flower. You can even kind of pull the petals apart to reveal the parts on the inside of the flower if you'd like. And hold them still with your finger. Yeah, I think that'll do. So one other thing that I at least feel comfortable to do is, since in those photos you might not be able to tell some very fine characteristics, and with violets, this is just violets so far. I'm not telling you to do this to every plant. I'm going to tell you not to do this to every plant. But with violets that are not in a heavily polluted area, this is in the woods, I'm going to get some fine details on this leaf. Okay, so what I can do by putting that whole leaf in my mouth is your tongue is actually sensitive enough to feel that this leaf is hairy. You can't see the hairs in the photos, but I can feel them on the leaf. So in the notes of that observation, I can say leaves minutely hairy on the upper and lower surface. I can say that. because I don't have a microscope out here to see them. But I can just write that in the notes. And I wanted to freak y'all out a little bit by putting a leaf in my mouth. Don't put every leaf in your mouth. Okay. Violets. Okay. I wanted to tell you about trees real quick. We're just at least gonna do some cursory stuff here. When you're observing trees, you know, bigger woody plants, you want to make sure you get the standard characteristics that you're going to observe on plants. So if it's got leaves, if it doesn't have leaves, you shouldn't be observing it. But when it has leaves, you want to make sure that you can get all of the characteristics of that leaf in your photos. So this one has these teeth on the margin. You want to make sure you can see those teeth in your photos. Make sure you get the underside and the top side of those leaves. You also want to make sure you get pictures of the bark, because the bark can be very distinctive on woody plants. One other thing you want to make sure that you do, and we already talked about this a little bit, is you want to make sure that you're using all your senses when you're observing things in nature. So it's not just sight, or when you're observing birds, sound, or frogs, or what have you. You can also use your smell, and I encourage you to use your sense of smell. So just like we did with the Florida Indies, and I happen to know this one has a smell, you know, I can crunch that up real close to my nose and take a big whiff, and that smells like almonds. So that's another type of note that you can include in your notes. So the last kind of form of plant I wanted to give you some guidance on are large trees. So again, the thing we want to make sure we do is get pictures of the bark if we can. So this has very distinctive bark for a tree. We also want to make sure that we get the leaves. However, this is January and most trees don't have their leaves at this moment. So one thing you can do is look around on the ground right beneath the tree. Don't go too far away from the tree, because you'll get some other stuff. But if you look right beneath the tree, a lot of times you can find the old leaves. So this is a very distinctive leaf. And you can take pictures of these and attach them with this, because again, we want multiple photos. I mean, this is an oak right here, so you can even try to look for an acorn. However, this is a species of oak. This is a species of white oak. Makes great big acorns, and the wildlife often gobble them up immediately when they fall. So it's probably a bit too late for that. But that would be the reproductive structure we would want. For trees, I'll say, you don't always have to have pictures of the reproductive structures. It's very useful. It's a lot better if you do, but a lot of trees are very distinct, or they're distinctive enough to where you don't necessarily have to have that. But you do have to have a leaf. And you want to make sure it's the right leaf, which might be hard for you, because again, if you go too far away from the tree, like even here, I can probably find some different leaves. So there's a leaf of a different species right there. Here's a post oak. Here's a water oak. So you want to make sure, to the best of your abilities, that you're getting the right leaf for the right tree. One more thing I want to warn you about, if you're in parks and natural areas, is they'll often have interpretive signs next to a plant. A lot of people, when they make those signs, don't necessarily consult botanists. They consult things like iNaturalist, which can be wrong. So this is not the right species for this tree. So if you do come to Petal River Park, it's not this. We don't even have this species in South Mississippi, so it's wrong. This is not Quercus bicolor. This is Quercus machowii. Okay, so just to show you that you can actually do this from anywhere, you can do it in the field, like as you're observing these organisms, or you can wait and do it, say, in your car when your phone dies and you forgot to charge it. So what you're going to do is you're going to download the iNaturalist app. It looks like this right here. You will click that. It will open with all of your observations already on there from previous times, but if you don't have any, it's not going to show any, but you'll just hit the add button right there. Go to upload photos. You'll have to allow the app to have access to your photos, but let's say we're going to record that salamander that we saw earlier. So I'm going to click all of those, and let's say I'm also going to upload that violet that we saw. So I'm going to click all of those photos, and I'll go to add up there at the top. And so it's going to show you this interface here. So you're going to highlight all of the photos of one thing and then hit this group button there. So that's grouped all of those 5 photos into this one observation. Then for the violet, I'm going to do the same thing and hit the group button. So it's going to show all of our individual observations there. You can upload, as far as I know, as many photos in one observation as you want. There is a limit to how many photos you can do at once total though. I think it might be something like 20 or 30, so you might have to do several rounds of this to get a bunch of observation at once. But we're going to import 2 observations there. So what you'll find is that a lot of the times it should automatically upload the metadata for your photos, so that includes the Latin long there, so you don't have to worry about adding that. For my phone, for whatever reason, it won't automatically upload the time, which is odd. It used to. So that's the correct date. We'll knock that back about an hour so that it's around the correct time. And I can add some optional notes there if I want to now, or I can go to it later and add those. I can make the geoprivacy open, obscured, which essentially makes polygon around your observation about 10 miles wide, so that no one will be able to see, unless you allow them to, exactly where that was done. There are a lot of reasons you might want to do that. So if you're on someone's private property and the owner is not okay with people in the public being able to access where an organism is, that's one reason you might want to do that. Another reason is that you might be observing an endangered species, and poachers are real. You don't want someone to be able to find exactly where this, say, rare orchid is, so you might want to obscure that. So it'll still show up on the map, just in a broad polygon, not a specific point. You could also make them private, but then I won't be able to see them, so don't make them private, okay? So I'm okay with these being open to the public, totally cool with me. I can use this identify button here if I don't know what I'm observing, or I can go to search if I do know what I'm observing. But let's just try this for fun. So I click the identify app. It's now running these photos against every, it's running it through all of the photos in its bank and comparing it. And so this first picture, it's telling us there's an 81.5% likelihood that it is that species, and that's pretty freaking good. Plethodon Mississippi. Like I said earlier, this is a Mississippi slimy salamander. So you can actually click these photos up there at the top and see for that second photo, it can't, for whatever reason, be too sure that that's what it is, but it is 100% sure that it is at least a salamander, which is fine. You can identify it as that. I can keep clicking through these to make sure that I'm getting the same answer. So this is saying it's in the genus Plethodon. That's pretty good. Click on the next one, see what we get. It's also saying an even higher percentage now, 86%, that it's that species. And this even closer photo, it's also saying the genus Plethodon. I do know that this is Plethodon Mississippi, so I'm going to click that. You can actually read about and see other pictures of these if you click the little information buttons next to these. And then when you're sure that you know, or at what level you want to identify this as, I know this is Plethodon Mississippi, so I'm going to hit the check mark right there. So it brings up that identification. We've got all of this done for now, so I'm going to upload it. And now we have to do the violet again. So that's the correct date. That's the correct time, just about. We'll say about 3:20. That should be about right. Geoprivacy open, organism is wild. The optional notes, I'll do that later when I have more time and I'm not in my car. So for the violets, come on. Boom, there we go. So it's saying I'm offline. I don't know why it's saying that. But genus Viola, it knows it's a violet. It's more than likely using location data to tell me that it's probably the common blue violet, Viola sororia. And then I'll click on these photos up here. So all it's telling me is that all it's telling me it knows is that it is in the genus Viola. It is a violet. That would be fine. I can leave it like that, and I can upload it as that.
An important thing here is that if it'll save this if it's not ready to upload them immediately, and it'll go through that. Okay. So these are uploaded now. So the one that's more broadly identified, genus Viola, I'm going to click on that, and it will bring up our observation. We can scroll through the photos we saw, and then I can see all of the activity that's on that. I can leave a comment on this. I can suggest a different ID, which is the scientific part of this app. So different people can also suggest different or more specific identifications for this observation. Like later, I went and found that this was indeed the common violet, Viola sororia. There we go. So I can click the check mark there. Would you like to suggest the following identification? Yes, it is Viola sororia. So I'm going to suggest that. So now it'll cross out the old identification and enter this new one. Yeah, and I just wanted to show you that. You can do that to other people's posts. If you know what a species is, but someone else didn't when they observed it, you can go and suggest the correct or a better ID for it. So that there's also a little bit of a social element to this app that I like a lot. You can favorite observations if you want from other people or from yourself by hitting that star there. You can edit your observations using the edit button in the top right. You can see details of this observation on the app by hitting details, and it'll show you the map and where you have observed other things. Yeah, it'll even tell you the data quality. So needs more IDs essentially to become research grade.
Your observations, it should be fairly straightforward. What you need to do first and foremost, this is the desktop version, and if you go to community up here and you go to projects, every single one of you need to make sure that you join the project, and you can go to the search bar right here and type in BSC 305, and it should be the only thing to pop up. Yeah, so Evolution BSC 305 course project. You need to make sure that you join this project. Because I created this project, I can't quite see exactly what you'll see over here, but the button should be around this area here. Yeah, so what's going to happen is when you join this group, any observations that you have that you take during the time of this class will automatically be uploaded into this, so you don't have to do anything extra to add them to this group. So for example, over the weekend, I added these. I probably eight or 9, it says 9 right there, observations. So once you're in this group, that's fantastic. Now you can go to your observations, and you need to make sure that you have some non-optional parts to your observation. So you need to make sure it has the correct date, observation date and time right there. You need to make sure that you have the Latin long displayed, so you can't hide any observations you make here, at least for the purposes of this class. You need to make sure that you have a note typed in right here. So you saw in the mobile app where to go to type those in, but if you want to wait and do that on the desktop app, you go to Edit on your observation right here in the top right, and you can add notes right here. You'll notice I have the species name right there between these asterisk marks. That doesn't do anything except make it italicized. So again, remember when we're typing species names, we try to italicize as much as possible. So there's no italics button on here, but if you just kind of insert it between two asterisks, it will automatically do that. So once you've done that, make sure you hit Save Observation, and it should add that note. Like I added a period at the end of that, so there it is right there. The next thing you need to make sure you have are annotations. So if you scroll down, you can see these herp people are kind of crazy. They commented immediately on it and confirmed that this is indeed a spring peeper. So you want to make sure you notate whether or not the organism is alive or dead. Sometimes, it's a pretty apparent in this photo that it's alive, but sometimes it's not always that apparent. Established doesn't really matter for our purposes, so you don't have to do that. Evidence of organism, that is a very broad category. You can actually get rid of any annotations that you've added. So if I accidentally clicked organism and I wanted to go back, I can just hit that X right there and it'll go back to a blank evidence of presence. So, these can look anything like bones or constructions like nests or whatnot, eggs, the organism itself, scat, so like that poop that we found in the woods, or tracks potentially. But this is the entire organism, so I'm going to click that. If you can tell what life stage it's in, we can we can notate that here, whether it's an adult, egg, juvenile, or larva. So I know this is an adult. And then if you're able to determine the sex of that organism, then that's appropriate. But if you can't, you don't have to worry about that. And the last thing I want you to make sure that you do is make sure that this is showing under that right there. So it should have this under projects. And really that's it. So like I said, for this class, you just need to make sure that so these are the 8 I observed the other night. Make sure that you have all of those for each of your 25 observations. Okay? And if you have any questions, of course, feel free to, come to office hours, ask me, or if you're on campus, feel free to come visit me in person.